I'm one of the group leaders in the Infectious Disease Initiative at Chan Zuckerberg Biohub, and we're located across from UCSF Mission Bay, just downstairs from BayViro. And today I'll talk about one story that we're close to wrapping up on the isolation and characterization of broadly neutralizing antibodies against dengue virus. So before I start, I'd like to emphasize that although this project has been largely driven by Natasha Durham and Aditi Agarwal in my group, this involves ongoing and future collaborations involving many individuals both within Biohub and also outside of Biohub, and I'll try to emphasize these individuals throughout the talk. Okay, so dengue virus is a member of the flavivirus genus, which is a group of about 70 envelope positive stranded RNA viruses. And this genus includes important human viruses, some of which are listed here. So these viruses are transmitted to humans by a mosquito or tick bite, and they cause a range of symptoms from a mild fever to severe neuroinvasive or hemorrhagic disease. And for many emerging flaviviruses, there are no effective vaccines or antiviral drugs. And bear with me, but just to get everyone on the same page, I'm going to go into some fairly detailed background on B-cell development and antibodies, because some of these details will be important later. So antibodies are Y-shaped proteins with two heavy and two light chains that are linked together by disulfide bonds. And the tips of this Y-shaped arms represent the variable or antigenic binding region, and the stem represents the constant or Fc region that's important for effector-mediated functions. And what I'll talk about today are neutralizing antibodies. So these antibodies can act without relying on Fc effector functions, and instead can directly inhibit virus entry by coating the surface of the virions. And the variable regions of the antibodies are assembled by a recombination of different gene segments. These are V, D, and J genes for the heavy chain, and V and J for the light chain. And within the variable domains, there are three regions of high sequence variability, and these are known as the CDRs. And a clonal family of antibodies are those that are encoded by the same V and J gene and have the same CDR3 length. And so upon antigen activation, B cells can undergo further diversification by somatic hypermutation of their antibody variable genes. So this process, known as affinity maturation, usually occurs in specialized regions within secondary lymphoid organs called the germinal center. So through this process, B cells that express antibodies with improved affinity for the activating antigen can be selected for further expansion and are then released into the blood as either plasmablasts or memory B cells. And following acute infection or vaccination, these plasmablasts transiently circulate in the blood before migrating into secondary lymphoid organs where they can further differentiate into tissue-resident plasma cells that are more long-lived. And so one thing to note is that plasmablasts in the blood can originate from either newly activated B cells or from circulating memory B cells. And because plasmablasts are generally generated during acute infection, more than 75% of them have been shown to be antigen-specific versus a much smaller percentage for memory B cells. And so the antibody response to dengue virus, and there are 4 circulating serotypes, the complex antibody response has made it challenging for vaccine development efforts. So although following primary infection, the antibody response includes potently neutralizing antibodies that are serotype specific, the majority of the response consists of cross-reactive but poorly neutralizing antibodies. And it's thought that after primary infection, individuals are generally protected from reinfection by the same serotype, but they remain at risk for secondary infection with a different serotype. And many decades of epidemiological studies have shown that secondary infection by a different serotype is a major risk factor for development of severe disease following dengue infection. And this phenomenon is not well understood, but it's hypothesized to be partly due to these cross-reactive, poorly neutralizing antibodies to enhance infection by promoting uptake into Fc receptor positive target cells. And this process is termed antibody-dependent enhancement, or ADE. So because of this phenomenon, it's thought that an antibody-based vaccine must elicit durable and potently neutralizing antibody responses against all 4 serotypes simultaneously. So all of the leading dengue vaccine candidates that are in clinical development are based on a tetravalent strategy, which relies on eliciting a balanced and potent polyclonal neutralizing antibody response against each of the serotypes. And ideally, the rational selection of strains to represent those that are circulating. And these two factors played a role in the poor efficacy of a recently licensed dengue vaccine. So this is the first and only licensed vaccine so far. Unfortunately, this vaccine failed to equally protect against all 4 serotypes, and reduced vaccine efficacy was observed when there was poor amino acid sequence match between the vaccine and circulating strains, and also among recipients who had no prior dengue infection. Presumably, vaccination in these previously seronegative population mimicked a primary infection that elicited poorly neutralizing antibodies with the potential to enhance subsequent infection. And in fact, long-term follow-up studies showed that within these subpopulation of vaccinees, there was increased risk of disease following subsequent infection. And although, again, the mechanism behind this phenomenon is unknown, ADE is hypothesized to play a role. And so the poor efficacy and safety profile of this dengue vaccine, despite many decades of research, highlights the challenges of eliciting protective antibody responses against dengue virus because of the risk of ADE. And so my research group at BioHub is focused on identifying host and viral determinants of antibody-mediated neutralization or enhancement. So when Caleb was in the lab, he studied the host factors required for ADE. He's currently at NGM Bio, but I think he's here in the crowd today. The focus of my talk today is actually on the study led by Natasha and Aditi on identifying determinants of neutralization. So the main target of flavivirus neutralizing antibodies is the envelope, or E protein, which mediates entry into cells and forms head-to-tail dimers consisting of 3 structural domains shown in the different colors. And the E protein is anchored to the viral membrane via this transmembrane and stem regions. And on the surface of the virus particle, the E proteins are densely arranged into 90 E homodimers. And this quaternary arrangement is important for antibody recognition. So excitingly, a few years ago, a group at Oxford and at the Pasteur Institute identified a class of broadly neutralizing antibodies that target an epitope spanning both monomers within the dimer, shown in green. And so this epitope is known as the E-dimer epitope, abbreviated as EDE, which is a little confusing. But there are 2 subclasses of these EDE antibodies. EDE1 antibodies can neutralize not only all 4 serotypes of dengue, but also Zika. And EDE2 antibodies have more limited activity against Zika virus. And none of the EDE antibodies can neutralize West Nile virus. So antibodies targeting this EDE epitope represent the only class of broadly neutralizing antibodies against flaviviruses so far. And as Rebecca nicely introduced, for other antigenically diverse viruses, I'll just take HIV as an example, there have been many more classes of broadly neutralizing antibodies identified. And the identification of these multiple sites on the viral glycoprotein that can be targeted by broadly neutralizing antibodies has provided a template for vaccine design. And so we're interested in identifying whether there are other vulnerable sites on the flavivirus E protein targeted by broadly neutralizing antibodies to inform vaccine design, and also how these broadly neutralizing antibodies evolve from the overall B cell repertoire. And our long-term goal is to inform epitope-based vaccine design as an alternative strategy to this tetravalent strategy by identifying a conserved site that can be rationally engineered into an immunogen to elicit a broadly neutralizing monoclonal antibody response against all 4 serotypes simultaneously, with the idea that this could potentially mitigate the challenging issue of strain selection for the vaccine. And so the broadly neutralizing antibodies that I'll talk about today were identified by following up on a now published study led by Fabio Zanini in Steve Quake's lab and Makeda Robinson in Shirin Ainov's lab. So this study involved single-cell RNA-seq of B cells from 2 individuals who were acutely infected with dengue virus, and they were enrolled in a Colombia cohort initiated by Shirin. And so Fabio and Derek in the Quake lab analyzed approximately 400 B cell sequences and identified 15 clonal families consisting of a total of 38 antibody sequences from these 2 patients. And besides being clonally expanded, we noted several other interesting features. First is that all the sequences corresponded to plasmablasts, which suggests a response to acute infection. Second, some of the sequences were extensively hypermutated, which indicates that these were affinity matured antibodies. And then finally, and most surprisingly, one of these clonal families had members that were found in both patients, which is highly unusual and is suggestive of convergent evolution, presumably to dengue infection. So to characterize these antibodies functionally, we then cloned the paired heavy and light chain genes into expression vectors, and this was done in collaboration with Crystal McCutcheon and her antibody discovery group at BioHub. And these antibody genes were then transfected in a 96-well format, and then secreted IgG was detected in the supernatant corresponding to 28 out of these 36 total monoclonal antibodies. And these antibodies were then tested for their ability to neutralize a panel of flaviviruses, including all 4 serotypes of dengue, Zika, and West Nile virus. So from this pilot study, we found that the majority of the antibodies tested, 25 out of 28, could neutralize at least 2 dengue serotypes, which wasn't all that surprising because these individuals live in a dengue endemic region. But 6 out of the 28 antibodies neutralized 4 different flaviviruses. And so as I mentioned, in these pilot studies, they were performed with crude and undiluted transfection supernatant. So for follow-up studies, we scaled up the production of these top 6 antibodies using purified IgG. And these were then tested in dose-response neutralization curves. So here I'm showing you a summary of the IC50 values of these 6 test antibodies as a heat map with a darker red color indicating increasing neutralization potency, and the gray boxes indicating no neutralization. As comparison, I've included the 2 subclasses of the known EDE broadly neutralizing antibodies and a West Nile specific antibody as a control. And so as you can see from the colors, most of these 6 antibodies displayed broad neutralization of dengue virus, but most of them with modest potency, including the 2 antibodies that were found in 2 different patients but belong to the same clonal family. And interestingly, this one antibody, I7, did not neutralize dengue 4, but neutralized the more distantly related West Nile virus with more potency than dengue serotypes 1, 2, and 3. But the antibody that we're most excited about is J9, which, as you can see from the color, unlike the EDE1 antibody, did not neutralize Zika. However, it neutralized the 4 serotypes of dengue with greater potency than the known EDE broadly neutralizing antibodies. And the reason we care about antibody potency against dengue virus is because flavivirus neutralization and ADE are governed by the number of antibodies that are bound to the virus particle. So this is most apparent when flaviviruses are used to infect K562 cells, which express Fc gamma receptor and are poorly permissive for infection in the absence of antibodies. So this makes K562 a good system to study ADE. So in this system, we see no infection in the absence of antibodies, but ADE is observed within a range of intermediate antibody concentrations, beyond which neutralization is observed. So over here, the number of antibodies that are bound to the virus is sufficient to mediate neutralization. So this observation suggests that even neutralizing antibodies have the potential to mediate ADE at subneutralizing concentrations. So consistent with this, antibodies that can recognize dengue have the potential to mediate ADE at varying ranges of concentrations. So among all the antibodies that we tested, including the 2 EDE antibodies, we found that the antibody concentration at which the peak level of ADE was observed was the lowest for J9. So this indicates that highly potent antibodies such as J9 are desirable because their potential for ADE is restricted to a narrow range of low antibody concentrations beyond which neutralization is observed. So one factor that can impact flavivirus, antibody recognition of flaviviruses is heterogeneity in the virus maturation state, which in turn contributes to structural heterogeneity. So flaviviruses bud into the ER lumen as immature, bumpy, non-infectious particles on which the E proteins associate with a chaperone protein known as PRM. So PRM is cleaved during viral egress, and this results in the formation of mature infectious particles with a smooth surface. And although PRM cleavage is a required step in the virus life cycle to produce these mature infectious virus particles, PRM cleavage is not required to be complete. And in fact, infectious viruses can be heterogeneous with respect to their PRM cleavage efficiency. And you can observe this from electron micrographs of a single flavivirus preparation, where we can see mature viruses, immature viruses, and partially mature viruses that retain some PRM. And the viro and maturation state can impact neutralization sensitivity. And in general, the presence of PRM increases the neutralization potency of most antibodies. And this effect is most dramatic for antibodies targeting poorly exposed epitopes, such as those within the fusion loop. These antibodies can only neutralize viruses prepared using standard methods, which retain some PRM, but not viruses produced in the presence of overexpressed furin, which increases the PRM cleavage efficiency. And importantly, a very recent study showed that dengue virus, isolated directly from patient plasma, are more mature, meaning they lack PRM on the surface, compared to those passaged just once in cell lines. And so although this study was only restricted to dengue virus serotype 1, it suggests that in order for antibodies to be effective in vivo, they have to be able to neutralize mature virus particles. So reassuringly, unlike fusion loop antibodies, which cannot neutralize mature viruses efficiently, both J9 and the EDE antibodies, their neutralization potency was not dependent on virus maturation state. So we next wanted to understand the mechanism of neutralization, which can happen at distinct steps during viral entry, including blocking attachment and theoretically reversing viral attachment to the receptor, or blocking viral fusion within the endosome. And the ability to block infection at a post-attachment step is a characteristic of most antibodies with potent therapeutic efficacy in animal models. And this could be partly due to the fact that no single receptor has been identified for flaviviruses, which instead use distinct cell surface attachment factors depending on the cell type. So it's possible that antibodies that can only block attachment might have cell type dependent activity. In addition, virus antibody immune complexes are taken up by Fc gamma receptor cells for clearance, so it's possible that antibodies that can neutralize at an intracellular post-attachment step might provide another layer of protection. So we tested the ability of J9 to block infection before or after viruses have been allowed to adsorb to cells at 4 degrees to prevent internalization. And we found that J9 and the EDE antibody could neutralize viruses, dengue virus potently in both assays, suggesting that they can act even after viruses have attached to cells. And this is in contrast to a control antibody that can only neutralize effectively before a virus has attached to cells. Next, we wanted to map the epitope targeted by J9, and to do this, we used 2 complementary approaches. The first is in collaboration with a company called Integral Molecular. So they generated a dengue virus 2 E-protein alanine scanning library for use in immunofluorescence studies to identify residues important for J9 binding. In parallel, because J9 could neutralize all 4 serotypes of dengue but not Zika, we introduced single mutations at a total of 29 solvent-accessible residues, shown in orange. And these residues are those that are conserved across dengue, but are different in Zika. So dengue virus encoding these E-protein mutations were then tested in neutralization assays. And so here I'm showing you the contact residues of the known EDE-specific broadly neutralizing antibodies as the spheres. So one monomer is shown in blue, the other in gray. And as you can see, the epitope of these antibodies span both monomers within the dimer. We also found that the 14 mutations that impacted J9 binding and or neutralization also span both monomers within the dimer. And in fact, 3 of these residues overlap those of the EDE broadly neutralizing antibody residues. But despite the similarity in the overall epitope targeted by J9 and the previously described EDE antibodies, we found that the molecular determinants of recognition were distinct. And in fact, a subset of mutations shown in the different colored spheres here had opposite effects on J9 and EDE, broadly neutralizing antibody activity. So here I'm summarizing the fold change IC50 values against the mutant viruses compared to wild type, with values above or below 1 representing decreased or increased sensitivity, respectively. So when tested individually, these mutations had modest or no effect or no reduction in J9 neutralization potency, but when tested in combination as double mutations, they had much larger effects, up to 100-fold reductions in potency. And interestingly, a subset of these mutations actually increased the potency of the EDE antibodies up to 100-fold. And however, these mutations had no effect on the EDE2 class of broadly neutralizing antibodies. So these results suggest that there are distinct neutralization determinants of J9 and the EDE broadly neutralizing antibodies. And the results here particularly suggest that the mutations that reduced J9 neutralization potency did not alter the overall antigenic structure of the virus. Next, to understand how J9 evolved from the overall B-cell repertoire, we collaborated again with Krista McCutcheon and Eric Waltari in the antibody discovery group. So they performed RT-PCR of B cells obtained from PBMCs of the J9 donor, and then performed next-generation sequencing. So I'll try to simplify this illustration here. This is a summary of a tree of members of the clonal family, which includes J9, and these sequences are derived from this common germline sequence. And as a reminder, a clonal family of antibodies are those that have the same V gene, J gene, and same CDR3 length. So each circle here represents a distinct IgG sequence, and the size of the circles represent the number of reads found for that sequence. And Krista and Eric analyzed 2 time points post-onset of fever for this patient, either 8 days or 22 days post-fever. And the numbers at the branches here represent the number of nucleotide mutations compared to the germline sequence. So from this analysis, we took away a few things. First is that J9 was derived from a membrane B-cell response because only highly mutated clonal variants were identified. And second, this clonal family experienced a large expansion. So there were 3 sequences that matched the J9 sequence exactly, 63 sequences total in this J9 cluster, and approximately 50,000 sequences in this family. And interestingly, when we tested the neutralizing activity of closely related J9 clonal variants, or a more distantly related variant, we found that all these antibodies had a very similar breadth and potency against all 4 serotypes of dengue virus. So to summarize, we've identified some broadly neutralizing antibodies against dengue virus from single B-cell transcriptomes of 2 acutely infected individuals. And among these antibodies, we identified J9, which was more potent against dengue virus than well-characterized EDE broadly neutralizing antibodies. And even though the epitope targeted is generally similar, the neutralization determinants of these antibodies are distinct. And I didn't really emphasize this, but EDE broadly neutralizing antibodies and J9 were derived from distinct germline sequences. And this is just to contrast with many HIV and influenza broadly neutralizing antibodies, which seem to display a restricted germline usage. And as I showed you in the last slide, even within the J9 clonal family, we identified broadly neutralizing antibodies with divergent evolution. So overall, these results suggest that there are multiple evolutionary pathways to generate flavivirus broadly neutralizing antibodies targeting a vulnerable site on the E protein, which could be good news for vaccine design to elicit these types of broadly neutralizing antibodies. And so we have some planned collaborations going forward. In the short term, we want to identify which somatic hypermutations are required for the breadth and potency of antibodies like J9. We also are interested in investigating the in vivo therapeutic efficacy of this antibody, and also determining the cryo-EM structure of the virus-antibody complex with these collaborators. So again, I'd like to thank both our collaborators within BioHub and outside of BioHub, and also the BioHub leadership team and the people who make sure the lab runs smoothly. And with that, I'll take any questions. Thanks for listening.
Individuals that you sequenced, that they had this, phenomenal, broadly neutralizing antibodies across all genotypes, or do you think that every acutely infected patient has buried some antibodies in their responses that might just not take over effectively enough to be conferring immunity?
Yeah. So I think it's been previously shown that the plasmablast response following repeat dengue infections do encode IgGs that tend to be broadly neutralizing. So it seems that there's sequential infection with different serotypes seems to broaden the antibody response. But there is actually something unique about the individual from which J9 was isolated from, in that she was enrolled in the cohort 4 days postpartum. So yeah, I'm not sure how that affects the antibody response. And I think related to your second half of the question, as you can see, J9 was not, even though it was part of a large clonal expansion, within that individual, this family was not the most prevalent. So one thing you might be wondering about, so the donor actually went on to have severe disease. If she had good antibodies, why didn't it prevent disease? It could be because it wasn't the most dominant response in the individual, and also from the epitope mapping studies, when we tested those mutations that affect potency of the monoclonal antibody, those mutations had no effect on the serum polyclonal response. So it's possible that this monoclonal response was not the most dominant in the overall polyclonal.